Hello guys, welcome to this new session on EST, that is Express Sequence Tags, and SNP, that is Single Nucleotide Polymorphism. So in this lecture we are going to discuss how these ESTs and SNPs are detected, what are they, and their application in the field of genomics. First, we will discuss about EST and then about the SNPs. So EST, that is Express Sequence Tags, it is a unique stretch of DNA within a coding region of the gene that is useful for identifying full-length genes and serve as a landmark for the mapping. And EST is also called as sequence tag sites, that is STS, which is derived from the cDNA. Now this STS is a short segment of DNA which occurs but once in the genome and whose location and base sequence are known. Now these ESTs or STS, they can be determined by or they can be detected by polymerase chain reaction, and they are very helpful in localizing and orienting mapping the sequence data and serve as a landmark in the physical map of the genome. So in the last year, you have learned what are the genetic map and physical map. So for physical mapping of the genome, these ESTs are very useful. So these ESTs are the small pieces of DNA. Particularly, they are being sequenced by using cDNA. So this short and single pass sequence, which are read from mRNA. So first of all, mRNA is isolated from the cells. Then they are converted into cDNA. And the short stretch of cDNA is sequence, which is now called as EST. So bits of DNA sequence, that is a short DNA sequence, it represents the gene expressed in single cell, tissue, or organ from the different organisms. The length of these EST is roughly around in between 100 to 800 nucleotides. And the application of EST is that we can identify the unknown genes and we can map their position within the genome. So if you have a whole genome sequence, but you do not know where the genes are, then we can simply determine the sequence of these ESTs, and with the help of these ESTs we can identify the position of the genes, that is unknown genes on the genome, as well as we can map their position on the genome. Also, EST provide researcher with a quick and inexpensive route for the discovering new genes. Now suppose you do not know the genes which are present on the genome, then we can use the sequence of EST to identify those new genes. Then it can be used for construction of the genome map. So we already discussed that physical map can be prepared by using this EST sequences, and we can characterize the expressed genes in the tissue or in the cell. So it's a very economical approach for identification, characterization of the expressed gene. Express sequence tag preparation. So this EST are prepared from mRNA, but mRNA are very unstable outside of the cell. Therefore, they are converted into complementary DNA. So the first step is isolation of mRNA and conversion of those mRNAs into cDNA. Now cDNA is much more stable, and importantly, because it is generated from mRNA in which introns have been removed. So the cDNA consists of a gene sequence free from introns. Now this cDNA represent only express DNA sequence. Finally, the sequencing of that cDNA is carried out, but this sequencing is done for the beginning portion of the cDNA only. So how these ESTs are prepared or sequenced from the cDNA? The 5' EST. In the 5' EST, it is obtained from the portion of the transcript that usually codes for the protein. Now these regions, they tend to be conserved across species and do not change much within the gene family. So you have this cDNA sequence, you only determine the sequence of first 100 to 800 nucleotide. That is called as 5' EST. So when you determine the sequence of 5' region of the cDNA, it will be called as 5' EST. Now this is the region which code for the protein. Now this region, as it code for the protein, it remain conserved within the gene family. So across different species, you may find out similar sequence. Then another type of EST is called as 3' EST. Now this type of EST can be obtained by sequencing only ending portion of the cDNA, that is 3' region of the cDNA. Now this 3' EST, they are likely to fall within non-coding or untranslated region. You remember those untranslated regions are also called as UTRs. So this 3' EST tend to exhibit less cross-species conservation than do coding sequence. So this particular 3' EST will only be present in that species genome. It will not be obtained in the genome of the other species in the similar gene family or protein family. Understand? And the beauty of this 3' EST is that the chances of occurrence of this sequence are only once in the whole genomic DNA sequence. Application of EST in gene mapping. So we can use this EST as a tool for gene mapping as well as gene discovery. Now the 3' EST, which is also called as STS, sequence tag sites. It serves as a common source, that 3' EST serve as a common source of STS because their likelihood of being unique to a particular species is very high. And STS is a short DNA sequence that is easily recognizable and occurs only once in the genome or in the chromosome. So it is easily recognizable and occurs only one time in the genome or chromosome. We can use those 3' EST, which are also called as STS, sequence tag sites, as a genome marker, and we can perform the physical mapping of the genome. Then ESTs can be used for gene discovery. They are a resource for gene discovery. Now, tools in the hunting for new genes involved in hereditary disease, this EST can be used for that. And EST are generated rapidly and inexpensively. Only one sequencing experiment is needed for each cDNA generated, and they do not have to be checked for the sequencing error. So this is very useful method in order to discover the new gene in the genomic sequence, because you just have to isolate mRNA, convert them into cDNA, and determine the EST sequence only once, and you will come to know that which is the location of the gene in the genome by simply uh, aligning the sequence of EST and genomic sequence. Now these EST sequences which are determined, they are stored in the database which is created at NCBI. In 1992, scientists at NCBI developed a new database designed to serve as a collection point for EST. Now, once EST that was submitted to the gene bank, it has been screened and then it is annotated, and it was then deposited to the new database which is called as DBEST. So in the last semester, you have learned bioinformatics, and in bioinformatics, you have learned that NCBI have several databases, and one of the popular database of NCBI is GenBank. But if you search the NCBI, they also maintain this EST database where all the EST sequences are stored. Now, how the annotation of the gene sequences is carried out by using this EST sequence data. Now, you have EST sequences either from the chromatogram, that is from the DNA sequencing machine, and from the databases. Now, you check for its quality. If it's a high quality sequence, then you use those ESTs for identification of the consensus EST sequence. So you perform the multiple sequence alignment of the similar EST, and then you determine the consensus EST sequence. Understand? If the sequence of the EST is low quality, then it is simply discarded. Or if the EST sequence is very short, then also it is discarded. Only high quality and sufficient length of the EST sequences are used for the generation of the consensus EST sequence. Now you have a consensus EST sequence. You can theoretically translate it into the polypeptide sequence. That is, you simply determine the all possible 6 reading frames and generate the putative peptide. The conceptual peptide or conceptual translation is carried out. Then this putative peptide, that is that we have generated by conceptual translation of the EST, it is matched with the protein database like SwissProt or UniProt KB. Now, if you find out a significant match, then the functional annotation of the gene is carried out. If you do not find out any kind of significant match with UniProt KB or SwissProt, then that EST is termed as a novel sequence with the unknown function. If it matches with certain protein, then that functional annotation is carried out for that EST, and then from that EST the gene is discovered and its functional annotation is carried out. So this is the entrees page. Actually, you can see there EST database is present. So you can type in your query in that search box and you can find out the sequences from the EST database. So this is how the result look like. This is very similar to your NCBI Gene Bank result, but the sequences are EST. This is EST sequence format, EST database sequence format, just similar to your Gene Bank file format that have accession number, EST, DB EST ID, and so on. Then limitations of EST. It is very difficult to isolate mRNA from some of the tissues or cell types. Then the important gene regulatory regions or sequences may be found within the introns, and they will not be represented in the form of ESTs because for the generation of the EST we are only using the exons. For most ESTs, there is no indication as to the gene from which it is derived. And lastly, because a gene can be expressed as mRNA many times, the ESTs ultimately derived from this mRNA may be redundant because the single gene expresses multiple copies of the mRNA molecule and you're generating ESTs from those multiple copies of the mRNA molecule. So your ESTs will be redundant. They will be repeated a lot of time. And finally, how to organize EST collection. So you can do the clustering, that is we can associate individual EST sequences with the unique transcripts or the gene. Then assembly, we can derive the consensus sequences from overlapping EST belonging to the same cluster that we can do with the help of multiple sequence alignment. Then we can do mapping, that is we can associate the EST with the exons in genomic sequence. Now for that purpose again the sequence alignment has to be performed of EST and genomic sequences so that you come to know that where this EST sequence is matching on the genome and we can do the mapping. Then interpreting, that is to finding out the coding region because EST is prepared from cDNA by using cDNA. So by identifying the location where the EST is matching with the genomic sequence, we can find out the correct coding regions on the genome. So that is what we can do with the EST collection data. Now these are some private EST databases apart from DB EST of NCBI. That is SNBI from South Africa, MIPS, Munich and SIB, that is Swiss Institute of Bioinformatics. Then there is a TIGEM from Italy, then there is a DOTS from Pennsylvania, etc. The next topic of discussion is single nucleotide polymorphism. The short form is SNP and it is pronounced as SNP. SNPs are the locations within human genome where the type of nucleotide present can differ between the individual. So maybe in one individual at a particular location, adenine is present. In other individuals at the same location, thymine is present. So there is a difference in between the nucleotides at a particular position in the genome sequence. So SNPs are most common type of genetic variation found among the people. At least 1% of population must contain the same nucleotide variation for it to be considered as a SNP. If that type of nucleotide variation is present in less than 1% of the population, it will not be called as SNP. Now, SNPs occur roughly every 300 nucleotide. And since there are 3.2 billion nucleotides in the human genome, there are approximately 10 million SNPs in the human genome. So that's a very large number. Now, this single nucleotide polymorphism, that is polymorphism, is in a general term, it means many shapes. And in terms of genetics, it means different types of nucleotide present among the individuals in the population at a particular location. Now, it is the ability to appear in the different form. That is another meaning of polymorphism. So, single nucleotide polymorphism or SNPs, they are most common type of genetic variation among the people. Each SNP represents a difference in a single DNA building block called as nucleotide. Now, in a DNA sequence, variation occur when a single nucleotide, that is A, T, C or G in the genome, differs between the member of the species. Let's take an example. Suppose in some individual the sequence is AAG CCTA, and in some individuals the sequence is AAG CTTA. So here there is a difference of single nucleotide, which can be termed as SNP. But remember, we can call it SNP only when this variation is present in at least 1% of the population. Understand? Look at this diagram. In some individual, the sequence is G in one strand and complementary C in another strand. In some other individuals at the same position, the sequence is A in one strand and T in another strand. So this is called as SNP. Rest of the sequence is similar. So in human beings, almost 99.9% bases are similar, but the remaining 0.1% bases makes the person unique. We know that there is a variation in the phenotypic characteristics among all the individuals in the population of human, and those variations are due to single nucleotide polymorphism, and they attributes different attribute characteristics and traits are developed due to that 0.1% differences, and that will decide how a person look or whether the particular disease will be developed in his or her body or not. Now these variations can be of three types. One is harmless, that is, there is no change in the phenotype due to that SNP. Some are harmful, like in diabetes, cancer, heart disease, Huntington's disease, or hemophilia. And some are latent, that is, variation is found in the coding and regulatory region. They are not harmful on their own, and the change in each gene only become apparent under certain condition, that is susceptibility to the lung cancer. Now suppose an individual is having some SNPs in the genes which are responsible for tumor suppression, or they are responsible for DNA repair mechanism, or they are responsible for regulation of cell cycle, then in that case, sometimes due to external stimulus, those genes undergo development of that particular disease condition. So some individuals are susceptible to the lung cancer when they're exposed to some kind of carcinogenic compound. So that kind of variation is called as latent variation. Then SNPs are found in both type of regions, that is coding region as well as non-coding region, occur with very high frequency, that is 1 in about 1000 bases to 1 in about 100 to 300 bases. But the range of the SNP or the frequency of the SNP is usually 1 in 300 bases. The abundance of SNPs and the ease with which they can be measured make this genetic variation significant. Now, SNPs close to the particular gene act as a marker for that gene. So again, in the physical mapping of the genome, we can use this SNP as a marker. Now, SNP in the coding region may alter the protein structure made by that coding region. Now, this you already know As in the case of sickle cell anemia, there is just a single nucleotide variation or mutation, and due to that, the whole protein structure got changed, that is of hemoglobin. Then SNP mapping. Now, SNP mapping is carried out by uh, sequencing the genomes of large number of people. So you collect the genomic DNA sample from different groups in the human population. Let's take the example of human. You collect the genome sequence from the different groups of the human population, and then you determine the sequence of whole genome. Finally, you compare all the base sequence of those genomic DNA sequences to discover the SNP. By after DNA sequence alignment, you will come to know that where are the differences in between the genomic sequences, and those are the points where SNPs are present. So you can generate a single map of human genome which contain all possible SNPs. Then types of SNPs. Now there are basically we can categorize SNP on the basis of whether they are present in the coding region or non-coding region. So on the basis of that, they could be non-coding region SNPs or coding region SNPs. If they are present in coding region SNPs, again we can classify them as synonymous and non-synonymous. Again, non-synonymous is of two types, that is missense and nonsense. So first of all, non-coding region SNP. Now, this is a segment of the DNA that do not comprises the gene and does not code for the protein. Second one is coding region, SNP. These are the regions of DNA or RNA sequences that code for the protein. Then for the coding region SNPs, there are again different types, that is synonymous. First one is synonymous. It is also called as silent. The SNP in which both forms lead to the same polypeptide sequence is termed as synonymous. Sometimes it is called as silent mutation. So the different nucleotides are present at that location, but the change in the nucleotide results in the coding of same amino acid. Understand? That will be called as synonymous because the amino acid is not changing. Because although the codons are different due to degeneracy of codon, same amino acid is being coded. So it will be called as synonymous or silent mutation. Then there is a non-synonymous. If different polypeptide sequences produce, then they are called as non-synonymous. So due to SNP, the codon got changed and the new codon is now coding the different amino acid. Now this is called as non-synonymous SNP. A non-synonymous change may either be missense or nonsense, where a missense change result in a different amino acid, while the nonsense change result in a premature stop codon. So you know all these things from the molecular biology. Then effect of SNP. Either it will be silent, means there will not be any kind of effect on the protein sequence or protein structure or protein function, or it may alter the function of the protein. Either directly, it will alter an amino acid sequence and thereby it will alter the function of the protein, or indirectly by altering the function of the regulatory sequence. So if the SNPs are present in the regulatory region and due to that the regulatory protein that is being produced, it has an altered function, so indirectly the protein function or the gene function which is controlled by that regulatory protein will be altered. Then the role of SNPs in disease predisposition. So all these common diseases, they are multifactorial, they are dependent on interaction of many genes, and the genetic difference between human population make one population more susceptible to the particular disease. Then SNPs and cancer. Now SNP in genes which are involved in DNA repair or drug metabolizing enzymes which are responsible for metabolism and detoxification of carcinogens can act as a cancer susceptibility gene. Now suppose the single nucleotide polymorphism is present in the gene which is responsible for DNA repair. Now if that SNP results into the non-synonymous mutation or non-synonymous SNP where the function of that protein is getting changed, in that case the mutated DNA will not be repaired, and that may lead to cancer. Or if the SNP occurs in the gene which is coding for the drug metabolizing enzyme, so that enzyme will no longer be functional. So if the carcinogenic compound comes in the body of an individual, then the drug metabolizing enzyme will not be able to metabolize that protein, or metabolize that carcinogen, and it may result into progression into the cancer. So this can occur through increased activation of chemical carcinogens and decreased ability of cells to detoxify and repair mutagenic changes. Then methods of identification of SNPs. Now there are two types of SNPs. First type of SNPs, they are known to us. They are already known. We just have to detect them. And second one is identification of new SNPs. So we are going to discuss how we can do that. First of all, detection of known SNPs. Now there is a gel-based genotyping method. So first of all, what do? You amplify the particular region of the DNA where you are looking for the SNP. So you amplify it with the help of PCR, and then you digest the PCR product. So if the SNP is present at the restriction site, then there will be two types of allele. One type of allele will have an intact restriction site, and after restriction, 2 or 3 or whatever, how many number of restriction sites are present, those many fragments will be produced. And if the sequence is mutated, if it contains the SNP and another allele is present, then in that case, the restriction site will not be recognized by restriction enzyme, and it will not cleave the DNA into the fragments. So that is one way of knowing whether SNP is present or not. Then oligonucleotide ligation assay that we have already discussed in our genetic engineering course, as well as in the advanced genetic course. So I have provided you the PowerPoint presentation of molecular diagnostic tools where we have discussed this oligonucleotide ligation assay. So you can refer to that. Then there is a mini sequencing. Mini sequencing we can do with the help of HPLC. HPLC determine the single nucleotide, whether any specific type of nucleotide is present or absent, that we can determine with the help of HPLC, that is called as mini sequencing. So these are the methods to detect the known SNPs. Then there is non-gel based high throughput genotyping technology. Previous technologies that we have discussed, that is PCR, then mini sequencing, there in those cases we have to run the DNA on the gel. But there are other techniques which are non-gel based and they are high throughput genotyping technology though. So you can determine the presence of SNPs into all the genes present in the genome. The first one is hybridization using fluorescence resonance energy transfer detection. Examples is Stackman probe genotyping or molecular beacons. That also we have discussed in the last semester. That is molecular diagnostic tools. And next is high density chip array. So this is the DNA microarray method where we can use those Affymetrix chip for the detection of the SNPs. Then identification of new SNPs, how we can identify the new SNPs. It involves two steps. First one is conformation-based mutation scanning, and then direct DNA sequencing. So conformation-based mutation scanning. In this method, we take the single-stranded DNA molecule where we have to check the SNP, we have to detect the new SNPs. Then we allow it to fold into the complex structure. So that single-stranded DNA will fold into the complex structure, and then we run it into the gel. Now the mobility of this single-stranded folded DNA, it will be affected by its shape or by its folding pattern or the complex structure that it have achieved by folding. Now each and every DNA with the different sequence will be folded differently, and accordingly its mobility will be affected. So if two DNA sequences has difference of one nucleotide, even their mobility will be different in the gel. So in this way we can determine the presence of SNPs in the DNA sequence. Then use and importance of SNP. Variation in the DNA sequence of human can affect how humans develop diseases and respond to the pathogen, chemical, drugs, vaccine, and other agents. So it is very important for us to identify the polymorphism in the human genomic DNA, to identify the different alleles present in the human population, because all these different DNA sequences are going to be responsible for response to the different response to the disease, pathogen, chemical, drugs, and vaccines. Then SNPs are also thought to be key enablers in realizing the concept of personalized medicine. So that we are going to discuss in next few lectures of pharmacogenomics. Then finally, SNP applications. First one is gene discovery and mapping, association-based candidate polymorphism testing, then in diagnostics and risk profiling, then the response prediction, that how an individual is going to be response to the particular drug or vaccine, then homogeneity testing or study design, that is whatever number of individual you are selected for the study, whether they are homogeneous or not, means uh, they all contain the same type of alleles or not. Then gene function identification can also be done with the help of SNP. So in summary, a single nucleotide polymorphism is a DNA sequence variation occurring when a single nucleotide genome differ between the member of a species. They can act as biological marker, helping scientists locate genes that are associated with the disease. And when SNP occur within the gene or in the regulatory region near a gene, they may play a more direct role in disease by affecting the gene's function. I hope you like the lecture. Thank you very much.